So I'm in the bioengineering department and neuroscience program at the University of Colorado Anschutz Medical Campus. And I'm going to talk about our work in extending two-photon microscopy in freely moving mice. So let me start this. Okay, so miniaturized microscopy is becoming sort of more widely used because it really allows neuroscientists to have the freedom to study animals in a way that they can't get with sort of head fixed imaging, where you have to basically hold the mouse's head under the microscope objective lens. And so this allows a way to actually study social behaviors, navigation, and additionally more complex behaviors than you can. So some of the first results that came out using these miniaturized microscopes was with the wide field Miniscope device that basically uses just one photon light and then collects the light back on a miniature camera. And so this is showing some work imaging in the hippocampus back in 2013, I think from Mark Schnitzer's lab. And so this is, a really interesting result because you can get sort of the full information, spatial location information in the hippocampus and find how the different cell networks respond to the mouse's position. Okay, so again, this is, the previous example was using one photon wide field miniscope, they're called, and so it uses a sort of LED as its light source in a diachroic and some lenses and images onto a CMOS camera. And this is good in a way, but effectively you don't get, if you're trying to image deeper in tissue and you want to know, detailed structures of what you're seeing, something like two-photon fiber coupled microscopes can be of great benefit. So this is an example of some work that's been done on designing two-photon fiber coupled microscopes. In this case, with the scanning element included in the head attached mount. So on the left, it's showing a scanning element, which is a small MEMS scanner, and then with a hollow core fiber for light delivery, and then a separate collection fiber bundle. Or on the right, some work has been done with using piezo scanners that can scan the fiber itself. Okay, so some of the benefits, again, this is showing an example of imaging in the olfactory bulb. So on the left is showing a picture of just a wide field image like what you would see from a miniscope. And on the right is showing a two-photon image where you get this intrinsic cross-sectional imaging along with this enhanced penetration depth in tissue. And that's because when you're doing two-photon imaging, you only get excitation at the very focus of your objective lens, where the intensity is highest, and you don't get any excitation outside of the focal plane. And so some of the advantages, again, include the fact that you're going to longer wavelengths of light, and so you have decreased light scattering, and going to even farther wavelengths has now been done to perform 3-photon microscopy at around 1300 nanometers as opposed to 2-photon, which is typically performed between 100 and 1000 nanometers. And so this allows your effective scattering length to be increased as you're going to these longer wavelengths. Again, you have to worry about, of course, the water absorption, and so not every wavelength is suitable as you go to these sort of higher wavelengths. Okay, so some of the benefits of 3D imaging, again, if you're doing 2-photon, you get very nice optical sectioned images. And if you can scan your focus axially, you can resolve lots of the detailed structure that you see in the brain. So again, this is looking at glomeruli in the olfactory bulb, and you see all of the different structures and different layers of cells in the brain. So 3D imaging is also important. So looking at different methods, when we were trying to determine how to build our 2-photon microscope, we investigated different types of methods to perform this 3-dimensional axial scanning. So one method that's very fast is this tag lens, so tunable acoustic gradient lens. However, it's large lens and requires a lot of power. Another type of lens that's often used in benchtop microscopes are pressure-driven lenses, like the OptiTune lens, but those require that you maintain the alignment for them to perform. So we looked at electrowetting lenses, and these lenses can be made very small and take low electrical power, and most importantly, they're not sensitive to the orientation of the lens or any type of vibration. So the electrowetting effect, it's an interesting effect that was discovered almost 100 years ago. So in this case, if you take a drop of water, let's say on a hydrophobic layer on top of a conductor, it will basically, due to surface tension, right, it'll have a certain contact angle on that hydrophobic surface. But if you apply a voltage between that droplet and the conductor, you get sort of a rearrangement of static charge, and you can effectively control that contact angle. So this has been an interesting technique that's been used for making a lot of devices, so including, including like tunable lenses, prisms, and doing things with droplets. And so you can use this electrowetting technique to create a liquid lens. And so in this case, you effectively have a cylinder, right, that has a electrode conductor and then an insulator, and then you have your droplet of water and then a hydrophobic liquid like oil. Okay, and so when you apply a voltage, it'll change this contact angle between the hydrophobic coating and that liquid. And so it can change effectively the curvature. So if these have different indices of refraction, you can then form an electrically tunable lens. And so this is an example. Our collaborators up at CU Boulder work on developing these types of lenses. And so that's what it looks like when you're changing this applied voltage. Okay, so for our design for the 2-photon microscope, we effectively decided to create our optical relay. So we had a fiber bundle where we deliver the excitation light, and then had a collimating lens and our electrowetting lens, and 2 additional lenses to form our objective. And so here, by controlling this voltage on our electrowetting lens, we could change, the focal distance. So just with that applied voltage. Okay. And this is just showing, so that in this demonstration, we used a commercial electrowetting lens. Okay, and so as a result of this, we basically, so this was Baruch Osbey, a student in my lab. He created this very miniaturized device with just a few lenses and the electrowetting lens, and packaged it into this 3D printed device. And it only weighs a little bit above a gram, so it's very lightweight. And it's designed so that you can attach it or detach it from a base plate that's glued to the mouse's head. So in our design, we actually didn't include the scanning element in the miniature microscope, and that has some advantages because it makes it much easier to build these devices. You don't have to worry so much about alignment, and it also makes them much lighter weight. So I think that there's some important advantages to doing that. So to do our lateral scanning, we used what's called a coherent fiber bundle. So this is used often in medical endoscopy applications where you have just this bundle of very small fibers, between 10,000 and 15,000 individual fiber cores. And it's called coherent because they're aligned so that the structure of your fibers at one end, this proximal end, is equal to, or is equivalent to the fibers at the distal end. And so in this way, you can move your scanning element away from being on the head attached device, so onto the other side of the fiber. And then you effectively just focus your laser like a typical confocal or any type of laser scanning microscope. And then you can put the end of this fiber bundle at the focus and then scan the light across to form your lateral image. So effectively, what do you get out of this? You get a very pixelated image because you only get intensity at those individual fiber cores. But if you design your microscope magnification, you can effectively get similar cellular level resolution. Okay, so the main problem, in doing two-photon microscopy with these fiber bundles, is that effectively you're trying to pass this very short femtosecond pulse through 1.5 meters of glass. Okay, so the main thing that's happening here is that you get dispersion due to that glass. So the pulse out of your laser is extremely short, so on order of 80 femtoseconds or so. To support that short pulse, you do have a certain spectral bandwidth in this case. And so if you come in with your short pulse, all of the wavelengths in your spectrum are lined up. You can think of it so that your pulse is as short as possible. But if you travel through glass, what you find is that it has different indices of refraction for these different wavelengths. And so what effectively happens is your blue light travels slower than the red light. So at the end of your fiber, you're going to have a very stretched out pulse. So it goes from, let's say, hundreds of femtoseconds to, from 80 femtoseconds up to hundreds of femtoseconds or a picosecond. Okay, and again, because of this effect where you have the blue light traveling slower, you get what's called positive chirp, where the red light arrives first, the red part of your spectrum comes first, and then the blue part comes last. Okay, so one way to compensate for this type of dispersion is to use what's called, well, different types of pulse compressors. So there's a couple types. The one that we used is a grading pair pulse compressor. And so what you do is before you send your light in through the fiber, you pass it through this grading pair, and it's set up such that you know, you get what's called anomalous dispersions. You get the opposite of what happens with glass. So the blue light arrives before the red light. And then when you pass it through glass, right, blue light slows down and red light goes faster, and you can set it up so that you get a very short pulse out. Okay, so this is this is how, you know, we initially set it up. So we had the grading pair sending our laser pulse in with the grading pair and then sending it through the fiber bundle. And this works all right if you have low enough powers. But as basically, if you're trying to increase the power of your light at the sample, you see that no matter where you set your grading pair separation, you can't retrieve a short pulse out. And if you look at the spectrum of the pulse, so that's given in this red, you go from your standard spectrum of around 10 nanometers, and as you're increasing the power, it starts to decrease. And so effectively, you don't have the spectrum to support such a short pulse. Okay, and this is due to the nonlinearity when you're trying to propagate these very high intensity pulses through a very small fiber core. And actually the you know, these coherent fiber bundles are probably the worst thing to try to use for this. But so effectively what's happening here, so when you're in the nonlinear regime, your refractive index is not just, you know, constant value, but it starts to vary with the intensity, the instantaneous intensity of your pulse by this nonlinear parameter. So so what happens here is that because your index is varying with the pulse, as you're getting, you know, if you're on the front end of the pulse, right, your index is increasing with time, and what that does is it shifts the spontaneous frequency of your pulse. So in this case, let's say we've got a pulse coming in that's as short as possible, so its instantaneous frequency is uniform throughout the pulse. So in this case, if you get what's called self-phase modulation, you increase or decrease your frequency at the leading edge of the pulse and increase the frequency at the trailing edge of the pulse. And so that's typically what happens if you send a short pulse through glass fiber, is you'll actually get sort of a broadening of your spectrum because you're creating these additional frequencies. But in this case, what we were seeing was spectral narrowing. So what happens here is that we're sending in a negatively chirped pulse. And so, you know, the blue light was arriving in front of the red light. And then what was happening is that when you were shifting the frequency due to self-phase modulation, you ended up decreasing the amount of spectral frequencies that you had. Okay, so to compensate for this, one technique that can be used is to effectively put in another fiber. So if we added in another fiber, a polarization maintaining fiber, right at the exit of our laser. So the light coming in from our laser was coupled into this fiber, and then we had self-phase modulation, but that broadened out our spectrum. And then you can put that light through a grading pair to make it negatively chirped, and then run it through the fiber bundle, and that will that will end up narrowing the spectrum. But if you choose the right settings, like length of your fiber and settings on your grading pair, you can basically counteract those two effects and get the same spectrum out. And in fact, you can also use this technique to even shorten your pulse if you want. Okay, so this is the experimental setup, and you'll see more of this when we do the lab demo. So we've got the home-built laser scanning two-photon microscope, and then we put in our polarization maintaining fiber that broadens the pulse, and then the grading compressor. Okay, so these are some of the results that you can see with this. So this was some of the first results of doing some imaging on a mouse and just testing that it in fact can do behavior tasks fine with this miniature microscope and the fiber attached. And so we can get the mice to basically move around and do different behavior tasks. And in this case, it was running on a little wheel. That takes it a while to start going on that. I need to edit this video to get to the part where it's actually moving on the wheel. So there it goes on the wheel. Okay. And so this is an example of so imaging, so GCamp6S imaging in the cortex. So this is just through a cranial window using the two-photon fiber-coupled microscope. So these are examples of the images that you can see. Okay, and then by adjusting your voltage on the electrowetting lens, you can change the focal position and actually acquire images in 3D. And with this design, we can adjust our focus by 200 microns. Okay, and so this is some results where we did some imaging and a freely moving mice recording at different locations in the motor cortex. Okay. And this is an experiment. We've been working on this some more, so trying to use the fiber-coupled microscope with implanted grid lenses so that you could image in deeper brain regions. And this is just some initial results on this. So this is a grid lens that's implanted in the piriform cortex of the mouse, which processes the odorant signal from the olfactory bulb. And so this is a situation with the mouse. It was in a cage with some unfamiliar bedding. And so this was imaging it while it was moving around in that cage. And we see a strong correlation with the GCamp activity as the mouse gets near the unfamiliar mouse bedding. Okay, and so some of the other benefits, ideally what we want to do, and we're trying to work to commercialize this with intelligent imaging innovations. But ideally, we could have something that would fit onto, a sort of a standard multiphoton microscope. So you could attach this onto the microscope you currently have, and then use it. And so with that, you can basically do what you could with any type of standard microscope. So you can do simultaneous two-color imaging just by using your detectors on your microscope. And another interesting thing you can do if you have this type of axial scanning is acquire images in a tilted plane. And so this is an example as you're raster scanning. If you're changing the voltage on your electrowetting lens, you can effectively change the tilt of your imaging plane. And so that can be good if you're not, or let's say that you can't get your grid lens exactly in the right place, you know, or it's at some angle. This allows you to actually image at a tilt. And another thing that you can do with this electrowetting lens is fast multiplane imaging. And so this is just showing a result that you can actually switch this lens between two focal planes rather quickly. And there's a recent paper that came out where they did this, so with an electrowetting lens and a MEMS scanner. So I think it was by Zhang et al. In Nature Methods. And so this is, you know, using this electrowetting lens, you can get very quickly acquire from multiple planes. Okay, and so, you know, again, imaging, two-photon imaging through these fiber bundles, it does have a lot of advantages because the actual head attached part can be made very light. You're not including that scanner. But some of the downsides is that these commercially available fiber bundles are not really designed for, you know, two-photon imaging. So what you see here, so this is looking at fluorescence through our fiber bundle. I don't know if you can see the individual cores in that bundle on your screens, but if you're using one photon excitation, you see it's, you know, very uniform transmission through each fiber core. Whereas if you now look at two photon excitation, and this is just looking at a blank fluorescent slide, you see that there's a lot of heterogeneity, so different transmission through the different fiber cores. And so as we're finding, what's happening is that these different fiber cores have different amounts of dispersion. So they appear to be have different dopant amounts in them. And what that does is having that, creating a fiber bundle like that is good because then you don't get core-to-core crosstalk. So if you basically got, you know, these two fiber cores next to each other with equivalent phase velocity, they'll be crosstalk. So they intentionally do this, but that makes our life harder. So, you know, we're trying to take a look at ways where we could reduce this effect. Okay. So on kind of another topic, so I've talked to you about how we've been using electrowetting lenses, so changing the voltage to change the focal length of our lens for axial 3D imaging. So another thing you can do with electrowetting lenses is make electrowetting prisms. So in this case, you can make basically a lens where you break the electrode in two. So on one half of that cylinder, you have one electrode and another is a different electrode. So that's this given by this left and right. So you can apply different voltages, right, to these different electrodes. And so what you could do, so at some angle or at some voltage, you reach the critical angle where your right, the surface is effectively flat. And then if you change the voltage left and right about that critical angle, you can tilt, just create a tilted surface. And so this can be used as a scanner. So it's a transmissive scanner. So you come in with light, and when it hits that surface, if the liquids have different indices of refraction, it'll deviate the light. Okay, so and with our collaborators, Juliet Gopinath and Victor Bright up at CU Boulder, they developed a electrowetting prism that could do this. And so this is looking at basically by changing the voltage across these two different electrodes, they could see a difference in scan angle of the laser. Okay. And so this could be, an interesting way to incorporate this type of scanner onto a miniature microscope, as opposed to, the other mechanisms, scanning mechanisms I had talked about, like MEMS mirrors or piezo scanners. Okay, so we did the first demonstration of this electrowetting prism. So in our lab, in our custom-built laser scanning microscope, we effectively added an additional relay where we imaged the electrowetting prism onto the galvo mirrors. And then we could actuate just one axis of the galvo mirror and then use the electrowetting prism to scan the other axis. So this is sort of looking at the light. So the galvo was providing the fast axis, the electrowetting prism was providing the slow axis. Okay, and the electrowetting prism doesn't have, right, it's not, you don't get sort of a smoothly changing scan angle with voltage. So on the right, you actually see this distortion, but just with sort of computation, you could correct for that. And so we were able to image, this is just an example of imaging hippocampal neurons labeled with GFP, just fixed cells. So on the left is the image just taken with the two galvos, and on the right was taken with the electrowetting prism scanner. Okay, and let's see. So I wanted to, finally, I wanted to talk about some exciting work we're doing where we're trying to develop a fiber-coupled STED microscope. And so this is work between my lab, Juliet Gopinath, and Mark Siemens, who's at University of Denver. So we were interested in developing this type of fiber-coupled microscope that could do this STED super resolution imaging, because it might be possible in the future if you could do this through a fiber, you could then have a miniature head attached microscope on your mouse and be able to actually look at subdiffraction limited features in the mouse. So it's kind of our very lofty goal of trying to do this. So so to just go a step back and explain STED microscopy. So STED microscopy is a super resolution technique where effectively you start out with your confocal excitation. So a standard laser scanning confocal excitation, you're limited by diffraction. So you can only get resolution according to how tight you can focus your light. So if you're scanning your light over a sample, you're going to excite multiple fluorophores, and then you'll see this smeared out fluorescence, and your resolution is determined by roughly the wavelength over 2 numerical aperture. So in this STED concept, which was developed by Stefan Hell, who won the Nobel Prize. So you take this shaped laser. So you have what we call the STED laser, which is shaped in this kind of donut mode at the focus, where there's no light at the center, and then there's light on the outside. And this STED laser is at a different wavelength that overlaps with the emission spectrum of that fluorophore. So what happens is you come in and you're exciting your fluorescent molecules, but with the STED beam you get stimulated emission. So So where the fluorophores see this laser, they basically emit a photon of that specific wavelength, and then using a filter you can block that, and you only get sort of the effective excitation or effective fluorescence from the very center where there's no intensity in that STED mode. So using this, it's called diffraction unlimited microscopy, because just by cranking up your STED intensity more and more, theoretically, you could get as tight of or small a resolution as possible, but realistically, your especially biological sample can only handle so much intensity. But effectively, with sort of practical values of your STED light, you can get resolutions down to 40 to 60 nanometers. Okay, so in looking at how could we do this through a fiber, so there's a way of actually using fiber modes and propagating your laser. So one way to do this is to actually excite these higher order fiber modes. So instead of just the sort of single mode that's propagating through the fiber, you can excite a mode that has these two lobes, let's say here, and another that has two, right, horizontally, so two separated horizontally, two separated vertically, and then the result will be basically a donut-shaped mode. So if these two modes that you are exciting are coherent with one another, the donut-shaped mode will only happen if they're maintained and they're perfectly coherent. And so this would not be good because, if we wanted to use this for a miniature microscope, the fiber will bend or there'll be temperature changes. And so, the coherence relationship between these two modes is going to change. So what we're doing, which was thought up by Mark Siemens, is to basically delay those two modes by the temporal coherence length. So if you do this, then no matter how much you bend the fiber, you're still going to end up with that donut intensity at the end. Okay, so we've done some preliminary work of STED through the fiber. So this is sort of the focus through a high NA objective of that donut coming out of the fiber, and it was suitable for doing STED. And we overlapped it with the excitation beam that traveled through the same fiber. And this is the result of the spot at the focus, so in X and Y, and also in Z. So one of the interesting things is you don't get, just with this donut mode, you only get enhancement in your XY resolution, and you don't get the enhancement in Z. And so this is just a demonstration of 100 nanometer fluorescent beads, so taken through this fiber. And we actually had both the excitation and STED propagating through the fiber, and the fluorescence was also collected back through the fiber and then on a APD detector. So this is the result we got with this. So an improvement in resolution over 2 times. And we could also image cell samples with the same fiber STED. And so this is an image of Alexa labeled tubulin. Okay. Okay, so I just wanted to make sure that I thank all of the people that were involved in this work. Particularly, the key people who were involved in the development of the miniature microscope are Baruch Osbey and Greg Futia, who are both graduate students in my lab. And Baruch is now at intelligent imaging innovations, and work on the electrowetting prisms and prism scanners was done by Omkar Supakar. And then Brendan is a student in Juliet Gopinath's lab, and then Stephanie Meyer in my lab worked on the fiber coupled STED imaging. And I have a picture of Connor. I didn't talk about his work, but he's there. Okay, so thanks everyone. I hope that sometime in the future, when we can actually travel again, you'd be interested in visiting us in beautiful Colorado and seeing the Rocky Mountains. So we have this sort of University of Colorado Neurophotonics Group where we're doing this type of work, and we're always looking for good students and postdocs who want to come join us and come skiing. It's different from Santa Barbara. You have to like skiing. Yeah, so thanks very much.